So we will be studying specifically the exocrine glands. What is present in the exocrine glands that is absent in the endocrine? Ducts. So exocrine glands are composed of two basic components. The secretory portion and the conducting portion, which is technically called the duct. Okay, conducting portion, duct. The one responsible for producing the secretions, secretory portion. So we will begin with the gland, the parotid gland. Okay. So now, those are the secretory portions of the parotid gland. That is where saliva is actually produced inside the parotid gland. Now, do not be confused because normally as we grow old, our parotid gland will become infiltrated by those cells. Identify the cells pointed with large clear cytoplasm, and then the nucleus is pushed to one side. Adipose. Okay. Now, our tissue of concern will be those pointed structures, the secretory portion. What is the specific name of the secretory portion of the salivary glands?
Serous.
Acini. Singular form, acinus. Or what is the other name for the acini? Alveoli. Okay, the secretory portions are called alveoli. Now, notice the cytoplasm of the epithelial cells is dirty looking. That is so because of the nature of secretion of the parotid gland, which is purely serous. What's the difference between a mucus and serous secretion? Where will you see more proteins? Serous. Proteins in the saliva will come in what form? Amylase.
Yes.
Enzymes. Okay? Enzymes. Now, the reason why those cells have dirty looking cytoplasm, it's because of the presence of the organelles responsible for producing the enzymes. Enzymes are technically proteins. Proteins are produced by what organelles? So remember that the enzymes are not kept inside the cytoplasm. They are released from the cell. So that specifically the enzymes are produced by, come on, what is responsible for synthesizing proteins which are to be released from the cell? Ano? Okay, rough endoplasmic reticulum. After produced in the rough endoplasmic reticulum, the enzymes are packaged in Golgi apparatus, and then afterwards they will become temporarily stored in secretory granules. So that's the granular appearance of the cytoplasm on the alveolar cells. What is the shape of the alveolar cells? So actually they are described as pyramidal in shape. So parotid gland can be easily identified because of the serous characteristic of the acinar cells. Okay? Now, another thing to remember about salivary glands. The secretory portions are grouped into lobules. So that is one lobule, that is another lobule, that is another lobule. How do you call the conducting portion? Ducts. Okay? There are two general types of ducts. Those which are present inside the lobules, they are called intra. Inside intra. And then those ducts located outside of the lobules, they are called interlobular ducts. So identify the structure pointed. Interlobular duct. How about that duct? Intralobular duct. Within the lobule, intralobular. Outside of the lobule, interlobular duct. What is the shape of the cells lining the interlobular duct? Cuboidal. Single or stratified? Stratified cuboidal. Now, do not be confused with the ducts. They will start as simple epithelium. Single layer of epithelial cells only. However, as the size of the duct increases, there will also be a corresponding increase in the number, in the layers composing the epithelium. So ducts can be simple or stratified. So however, at this level, you should be able to identify that duct inside the lobule. It should be called intralobular. How about that duct? What is the nature of the secretion of this gland? Serous. Identify the organ. Parotid gland. Organ is parotid gland. So next is another type of salivary gland, the sublingual gland, because it is located underneath the tongue. Now, unlike the parotid, which is producing purely serous, sublingual will have a mixed type of secretion, mixed serous and mucus. But before that, identify the structure pointed. Is it inside a lobule or outside of a lobule? Outside. So that they should be called interlobular duct. So, a while ago, we have studied the appearance of the serous acini. So now, what will predominate in this histology will be the mucus acini. Can you still remember the appearance of the goblet cell?
Yes.
Okay. Imagine the goblet cells arranged side by side with each other. That should be the appearance of what type of acinus? Mucus acinus. So, close up view. So, tubular pa rin ang shape or alveolar. And then, the acinus will be composed of mucus-secreting cells arranged side by side with each other. So identify the structure outline. Mucus acinus. So there are many mucus acini present in that portion. How about that structure pointed? Were you able to see lumen in the serous acini that we have studied a while ago? Wala. Diba? How about that structure possessing a lumen? And then it is inside the lobule, intralobular duct. Okay? Intralobular duct. Unfortunately, the stain of this slide is very faint already. So there are many intralobular ducts. And then that structure pointed is mucus acinus. Another salivary gland, submaxillary gland. The stain is also very faint. So we are seeing acini with dirty looking cytoplasm. So that those acini should be serous acini. Okay? So mga acini, close up view, you will not be able to see any lumen. Okay? Pag mayroong mga granules and dirty looking ang cytoplasm, serous acini. Identify the structure pointed. Interlobular duct. How about that structure?
Intralobular duct.
Okay. Next slide is the ileum. Already studied this slide. So identify the tongue-like structures. Villi. What type of epithelium? Simple columnar epithelium. Now, as you can notice, the surface of the epithelium is not flat. Because number one, of the presence of the villi, and then the epithelium invaginates towards the lamina propria. So that is now, the invaginations will eventually form the small intestinal glands. So the pointed glands are called small intestinal glands. What is the other name of the glands of the small intestine? Crypts of? Okay. Crypts of Lyrecon. Which are lined by what type of epithelium? Simple columnar epithelium. So what is the classification of the small intestinal glands?
Simple tubular.
Is it branching or not branching? Not branching. And then what is the shape?
Tubular.
Tubular. So,
Simple tubular.
simple tubular gland. So now, for next slide, colon or large intestine. Are you able to see any bilus? Wala. So that should be your identifying feature. Major difference between the histology of the small and the large intestine. You will not be able to see any bilus in the large intestine. However, the large intestine will be lined by what type of epithelium still? Simple columnar epithelium. Okay? Simple columnar epithelium. Identify the cell pointed.
Goblet cell.
And then just like in the small intestine, the epithelium of the large intestine invaginates to form the large intestinal glands or the colonic glands, which are still lined by simple columnar epithelium. So based on configuration, how will you classify the glands of the large intestine?
Simple tubular.
Simple tubular. Next slide is human brown skin. There are two types of glands present in the skin. What type of epithelium?
Terrified squamous epithelium.
Okay. So the glands present here are the sweat glands. The sweat glands are located in the deep dermis. Okay. Now, are the sweat glands straight? No. However, what is the shape? Tubular or alveolar?